Welcome to our podcast on the nervous system. This is for lab, and so we're going to look at a lot of histological images of different areas of the central nervous system, peripheral nervous system, and nerves in general throughout the body. In this first image, we're looking at a low magnification micrograph through a peripheral nerve, and you're seeing a bundle of axons, and you're seeing them bundled together into fascicles. And so with this one, we're going to look at higher and higher magnification as we go down, but basically what we're seeing is the epineurium that surrounds this entire nerve, and then the bundles of axons that are present within each. Now, in this particular image, we can see several nerve cell bodies. These are going to be our likely going to be either our sympathetic or parasympathetic autonomic nervous system ganglia. So we might imagine these being in one of our collateral ganglia, or they're going to be present in our dorsal root ganglia. You can see the characteristic nucleus, dark nucleolus, looks like an eyeball. And then many of the other cell nuclei that we see, much smaller, they're going to be neuroglia. And since we're in the peripheral nervous system, by and large, those are going to be Schwann cell nuclei. Here we're looking at more tissue. We see some blood vessels, we see muscle, but in the inset, we're going to be looking again at nerves, a bundle of nerves. And now as we look at this particular fascicle, what we're seeing is individual axons, and we can see that they're rimmed with this darker purple tissue. These are going to be myelinated. We have some blood vessels in this as well. So these are our myelinated axons. This connective tissue surrounding each is going to be our endoneurium. This entire fascicle has the perineurium, but it also is functioning as the epineurium, just like we saw with our muscle, the paramecium and the epimecium. And then we're going to have some areas where we have axons that aren't myelinated. Remember, those are also going to be supported by Schwann cells. They simply will not have undergone the process of myelination. Now, once again, some of these illustrate that this outer connective tissue is in fact epineurium, but where does the perineurium stop, the epineurium start? I never like to ask that unless we have several fascicles and we have the epineurium surrounding the entire nerve. Here we have a peripheral nerve in cross section. We have the nerve, we have some muscle, we have some tendinous material, and we're going to have the perineurium. This is going to represent one fascicle surrounding this entire structure. So as we look at higher magnification, again, we see the wavy nature to this tissue. When you look at that, you might be thinking, well, sometimes dense regular connective tissue can look like that. In this case, we got dense regular almost cut in a transverse section. You can see how tightly packed those are. These are fibroblasts. Certainly you should be able to recognize this tissue by now. This is our skeletal muscle. And this is going to be our perineurium, and they will say epi as well, but really one fascicle. I'm just going to call this peri. But as this waves and you see this lighter color to this, and these nuclei are somewhat elongated in nature, they're kind of fatter than what we'd expect to see for fibroblasts. It gives you the indication that something else is going on. And then you see these structures that even at this low magnification look, again, they look like butterflies to me. And when you see those at the light level, you know you are looking at nodes of Ranvier. And that's going to clue you in that you are looking at nerve tissue. Now, this is a much higher magnification, and we can see those little butterfly structures that we see here. But then we have these bubbles all along the length, especially we have these larger spaces that we see here. We have some spaces here, almost look like arrowheads or points. These are called Schmidt-Lannerman clefts. And these are places where as the myelination is occurring, most of the cytoplasm is getting squeezed toward the periphery, But if not all that cytoplasm gets out and the wrapping continues, you're going to have cytoplasm trapped in between these layers, in between the major dense line and the minor and the interperiod line. And these are called Schmidt-Lantermann clefts or enclosures. And that's what those in fact are. Now we're looking at a silver stain of a nerve. We've got a lot of nerve fibers there. Most of them are going to be myelinated in nature, and so let's look a little bit higher magnification at what we're looking at. Now in this particular stain, the myelin is a dark black, and so you can see how deep and intensely stained that is. And since we are looking at individual axons, then once again our endoneurium is going to be between. This will represent our peri and epi kind of collectively together, since we're working with just one fascicle. A collection of nerve cell bodies in the peripheral nervous system is a ganglion. And so we are looking in this particular image, I believe it is a dorsal root ganglion. And we're going to look at some of the cells that are present here. There are large pseudo-unipolar cell bodies. Again, nice big nucleus, prominent nucleolus. We can pick those out pretty quickly. We can see some of our myelinated axons that are going back and forth between. Almost looks like maybe a node of Ranvier. Those may be some Schmidt-Lantermann clefts, probably a node in that location. And a lot of the cells that we see surrounding these cell bodies are going to be what some call satellite cells. You see how they're circulating around these particular nerve cell bodies, but these are also Schwann cell nuclei. They're not myelinating the cell body, but remember, Schwann cells are going to support even unmyelinated fibers. Again, taking another look at myelinated axons. As we're looking through these images, we see our Schmidt-Lantermann clefts. We see our nodes of Ranvier. We've got some blood vessels here, and I'm not sure I see anything else really to mention. Okay, now looking at different organs, we have nerve cell bundles are going to be scattered throughout the body. So we'll see them in a lot of our tissues and organ histology that's upcoming. This is going to be peripheral to the prostate gland. And as we look at some of these materials, what we're going to be able to see, again, very recognizable, prominent nucleus, nucleolus, and we have a collection of nerve cell bodies in the peripheral nervous system. It is a ganglion. Here we see that wavy light appearance, and these are going to be our axons. Here we see a blood vessel. We've got red blood cells within that. Here's another axon bundle, fascicle of nerve processes there as well, but this is our ganglion. Now, in the intestinal tract, we're also going to have bundles of nerve cell bodies, ganglia. They're going to be present in two different locations. We're not going to worry about those. We'll cover those when we get to the alimentary canal. But again, when we look at them in histological preparations, again, we're going to be able to recognize them as our clearly visible nucleus, prominent nucleolus. And we're going to know that these bundled together masses are going to be our ganglia. Now, what we're going to have is we're going to have some associated with smooth muscle. These are our smooth muscle layers. This is going to be our circumferential layer. This is a longitudinal layer. And so we have some of these nerve cell bodies present here that are between the muscles. We're going to call this a myenteric plexus. And then this is going to be in the area that is just beneath the mucosa layer or the epithelial layer. As we get to organ histology, we call that the mucosa. And so this is going to be a submucosal plexus. But again, we will cover those when we get to the alimentary canal. Here again, we see our myenteric plexus. We have our circumferential smooth muscle, transverse section smooth muscle, large prominent cell bodies present in between. This is going to be our myenteric plexus. And again, this is going to be submucosal, large nerve cell body, very, very prominent. I mean, when you have the eyeballs looking at you, then you know you're looking at a nerve cell body. Now, looking further, we are now in spinal cord. We can see our white matter and gray matter, and the colors are almost, it's almost like an X-ray or a negative image, because the white matter is darker in this particular image, and here we have the gray matter on the inside. Here we have our commissure, dorsal horn, ventral horn. This is where we have sensory input, and this is where we have motor output with our motor nerve cell bodies. Now, as we look at sort of your very typical central nervous system tissue, we can see our nerve cell bodies. We can see the dark staining Nissl bodies present inside here, large nucleolus in the nucleus. And then this material that is surrounding it, made up of fibers, made up of microglial cells. This look, where you see a lot of space and a lot of fibers, is just kind of generically referred to as neuropil. So this is Nissl on the inside, and this is neuropil on the outside. Remember our term ground substance kind of was stuff. Well, neuropil is a sort of a generic term that means neuronal stuff. Now at higher magnification, we're looking at our neuropil. We can see our nucleus, prominent nucleolus, all of our Nissl bodies, and there are going to be some darker staining material that we can see present in these that are lipofuscin granules, but really collectively we just refer to that all as Nissl. Once again, very similar image, neuropil, neurons, prominent nucleolus, Nissl bodies. See some capillaries scattered out throughout there as well. Now at the EM level, I think it's fairly obvious that we're looking here at several myelinated axons. That's what these dark, thick rings are. But what we're seeing here is the nucleus of a Schwann cell, and it is actually associated with several different axons. And I know you're probably thinking, well, Schwann cells just myelinate one axon. It's the oligodendrocytes that myelinate several. That is correct. However, we have not myelinated these axons. These are unmyelinated fibers here. So this Schwann cell is simply associated with and supporting these axons, whereas this particular axon is heavily myelinated. And Schwann cells do both in the peripheral nervous system. Here we're looking at a higher magnification. Again, we can see our very prominent major dense lines. Really can't see the interperiod lines in these, but here is an axon, and it's associated with the cytoplasm of this Schwann cell. Now in this image we have a Schwann cell that is actually myelinated. Here we can see the nucleus of the Schwann cell, its cytoplasm, and here it is wrapping around this particular axon. So this is a myelinated axon in the peripheral nervous system, myelinated by this Schwann cell. Now we can very easily see our major dense lines. Again, the intraperiod lines are very difficult at best to see. And in this particular image, we can see the internal axon. And we can see at this point, we can see where we sort of stop and we have these sort of enlarged places, a little bit of cytoplasm. This is our internal mesaxon. And then out here where we can see this separation, this is our external mesaxon, the cytoplasm of the Schwann cell associated with the myelinating layer in here. And inside we have a little bit of cytoplasm in that first area of starting where the cytoplasm and the plasma membrane of the Schwann cell is inherently adjacent to the axon itself. This is simply the cytoplasm, the Nissl bodies, and this material that we see inside of a neuron. We can see a Golgi apparatus down here. These really dark staining, this dark staining material, these are these lipofuscin granules, but most of the rest of what we see is going to be our Nissl bodies. Once again, another view of a myelinated axon. We can see, again, mostly major dense lines, internal mesaxon, external mesaxon. Here we have a Schmidt-Lantermann cleft. There's a little bit of a Schmidt-Lantermann cleft. And we actually have collagen fibers out here. We've cut through a couple of mitochondria in the axon of this particular neuron. Now, we've seen this one in our lecture podcast, but this is where we see the very dark, intense lines, the major dense lines. And then you have to use your imagination a little bit, but almost the dotted line that you see running between, those are going to be the intraperiod lines. The dark lines are the two cytoplasmic faces of the plasma membrane together, and the intraperiod line are the two extracellular membranes that are opposed. Now, from our lecture podcast, you should easily recognize where we are in this particular organ histology slide. You shouldn't take a moment's hesitation to know. You see three layers: a sparse outer layer, very highly populated inner layer, and then this interface layer with these huge flask-like cells. What are we looking at? This is cerebellar cortex. And we have the molecular layer, the granular layer, and then these are the Purkinje cells of the Purkinje layer. Those help identify this as cerebellum. Now, I'm sure you know what this is, and I'm sure you realize it's not going to be easy. However, when we look at our 6 zones out here, as we're adjacent to the pia mater, which is on the surface, this is going to be our molecular layer. And you can see how this looks granular, right? This is our external granular layer. So we've got those two. Now, as we look, starting from this direction, it's almost like working a geometry proof for me. As we start looking at this layer, we're looking for some of the biggest cells. And as we get to these, we're going to know that these are going to be our giant pyramidal. So this is part of our internal pyramidal layer here. And so internal pyramidal is here. Then we've got our multiform layer down here. That means between our pyramidal and our granular, we have 2 more layers. And so as we look, you can see, I think that cell right there, you see how that almost looks pyramidal? There's one that looks pyramidal, there looks pyramidal. So we have molecular, external granular, we've got external pyramidal. Then somewhere in here we're going to have another granular layer. Do you see how this looks almost more like we have more cells and they're smaller than we have here? So external granular, external pyramidal, internal granular. Then we're going to get down in here to our internal granular, internal pyramidal, and then we'll have multiform, and by the time we get down to here, we're in our white matter. I think this sort of is curving around like this. This is going to be white matter down here. This is going to be multiform. Here we have pyramidal through here, internal pyramidal, internal granular, external pyramidal, external granular, and then we have our molecular layer on the outside.